last system, not system, but chapter for exam number 3, and then give us a chance to review. Now, if you remember, whatever we're reviewing today, whether we're going through exam objectives or just answering questions and things like that, we already have previously recorded exam 3 review sessions on Panopto 2. So those, I think the one that's on Panopto is maybe even like a full classroom session, like we had the whole hour and 15 minutes to review. So do check out the recordings as well. All right. So a key here for this last part of this chapter, we've been talking about how we regulate fluids in the body. We've been talking about electrolytes, but couched within electrolytes includes different things like acid and bases. So an electrolyte overall is something that when we put it into a solute, it falls apart. It dissociates into ions. Now, in the middle section there of this chapter, we were talking about salts. Salts that do that are like sodium chloride, potassium chloride, things like that, right? When those fall apart, we get ions, and then we talked about how we regulated certain ions, like sodium and calcium and potassium, etc. Now what we're talking about is a molecule that does the same thing. It's technically an electrolyte, but the difference between a regular electrolyte, like a salt, and something that is an acid or a base, is that when it falls apart, it is going to affect pH by either releasing hydrogen ions or binding them up, okay? So that's a key aspect. So yeah, we're still releasing ions because that's the definition of electrolyte, but one of those ions is a hydrogen ion, and that affects pH. And we know for things like enzymes, etc., it's critical that we maintain a certain range of pHs because that can affect body processes. So normally, our fluids will have, like we said, kind of an average pH. So for instance, arterial blood is about 7.4. Venous blood is about 7.35. And intracellular fluid is basically, and I say basically, although it's neutral, neutral. Right? If we remember, we've got a scale that goes from 0 to 14. So the halfway mark for that is 7, right? On our pH scale. And we have the smaller numbers here that are going to be more acidic, and the larger numbers here that are going to be more basic, also known as alkaline. And the reason that the smaller numbers are more acidic, this means is that we have an increase in our free hydrogen ion concentration. That means there's more hydrogen ions floating around. It has to be by themselves floating around. If they're a part of a molecule, that's not making it more acidic. It's only if the hydrogen ions are floating around on their own. In terms of basic or alkaline parts of this, often this means we have an increase in other things that are going to bind up free hydrogen, like hydroxyl, right? So when we're here in a pH of 7, water, pure water, which is our universal solvent, right, is actually neutral. It's not acidic or basic, because what does water break down to? It breaks down to a hydrogen ion plus a hydroxyl ion. So we've got just as many things that could bind up hydrogen as number of hydrogen ions that are floating around. Okay, so that makes it neutral. If we have more free hydrogens floating around, it makes it more acidic. If we have more things that can bind up free hydrogen or less free hydrogen floating around, that makes it more basic or alkaline. Okay? Also remember that each of the different steps along the way here, and those are not evenly spaced, but take it like it is, each of those steps along the way are a difference in 10 times the free hydrogen ion concentration, okay? Each step, it's a log scale. So when we're talking about pH, pH is calculated basically as 1 over the negative log of those hydrogen ion concentrations. Now, you don't have to worry about calculating that, anything like that in our chemistry class. But the point is, look at this, right? So number 1 is a log scale. That means every one number change here is a difference in 10 times that concentration. So if we move from 7 to 6, this has 10 times as many free hydrogen ions floating around in it. Down to 5, now it's 100 times more free hydrogen ions floating around. 1,000 times, 10,000 times, etc. Right? So the differences that we see in the numbers on the scale, even though it seems small, you're like, 7 to 7.35, right? Those are basically the same. Not the same. And in fact, small changes, what we see as small changes in pH can have large effects in the body, okay? And if we look at this equation, given that it's the inverse, right? It's 1 over that concentration. That also helps us to understand why it's the smaller numbers that are higher hydrogen ion concentrations. Because as this number gets big, this overall number for pH gets smaller, right? The biggest number you can have right here is 1 over 1. If we have 10 times that, if we have 10 free hydrogens, now it's 1 over 10, only 1/10. If we have 100 free hydrogens down here, now it's 1 over 100. That's even smaller, right? So the more free hydrogen ions we have, the smaller this fraction gets, and therefore the smaller the number gets on the pH scale. Okay? So again, don't worry about this. Don't worry about it at all. In fact, I don't know, there may be some part of my equation there that's wrong. I don't remember. But that does help us to remember that the smaller numbers on this scale are more acidic, the larger numbers are more alkaline, and it has to do with free hydrogen ion concentration. Okay? Okay. So, we need to maintain it within a certain range of pHs. So in fact, when we're talking about the blood, if the blood even gets like 0.5 100ths, right? If we're calculating decimal points, that's 0.5 100ths. If it gets even 0.5 100ths above the normal level for pH of the blood, you go into what's called alkalosis or alkalemia. It's too basic, right? If it gets even 0.05 on the pH scale below what it should be, you go into acidosis or acidemia instead. Right? So it doesn't seem like a lot, but do remember, right? It's a significant jump at each number on that scale. Okay, so where are these hydrogen ions that determine our pH? Where are they coming from, right? Why are we getting a bunch of them floating around on their own or being bound up, etc.? There's a lot of different physiological processes that affect pH, and this is where it kind of comes together as we've been learning about these processes in the stomach where we're producing hydrochloric acid, in the respiratory system where we have bicarbonate ions that are carrying around our carbon dioxide, and we're doing this conversion all the time in the red blood cells, in the urinary system where we know that we can secrete hydrogen ions and reabsorb bicarbonate ions, stuff like that. There's a lot of processes that are going on. Even with fat metabolism, remember when we were breaking things down, right? One of the byproducts, one of the products of the digestion of fats is we get fatty acids. When we're breaking down proteins, we get amino acids. When we're talking about our DNA, right, that's deoxyribonucleic acid. So there's a lot of important molecules that can be acidic. And so we need to be able to, when we're making those or breaking those down or something like that, it's going to affect overall pH, and we have to control that in some way. Okay? So there's a lot of different reasons why our pH might vary and why we might need to control it. Even this one, right? Everybody's heard of lactic acid from anaerobic respiration as well. Okay, so we need to be able to control that so that we don't get big swings in pH. Minor changes in pH are okay, but we don't want to get out of our homeostatically maintained range, right? So that means that if free hydrogen ions show up, we need to be able to bind them up really quickly so that we don't have a big change in pH. Or if we're going the other way, if it's becoming too alkaline, alkalosis is just as bad, then we need to be able to release hydrogen ions from somewhere as well. And that is essentially the role of a chemical buffer. A buffer is some sort of molecule, generally, that has the ability to either release hydrogen ions or bind it up. And that can help to control big changes in pH. It's a bit like when we were talking about hemoglobin, and we're like, okay, hemoglobin also needs to bind something up and release it at certain times. That something is oxygen. We don't want to hold on to it all the time. We want to be able to bind it up sometimes and then release it at other times as necessary for the body, right? And interestingly enough, hemoglobin also can bind up hydrogen and release release it. So once it's released that oxygen molecule, it can actually grab onto a hydrogen ion and help to stabilize pH. So it is part of our first of 3 different ways that we regulate pH in the body. It is part of the chemical buffer system. So again, these are different chemicals that we have in the body that can either bind up hydrogen or release them as necessary. So in terms of controlling our pH, we have 3 different mechanisms. One are chemical buffer systems, a certain number of things that can do that, and acting very quickly to grab up those hydrogen ions or release them as necessary. Second mechanism is the respiratory mechanism, and this basically involves, like we were learning before, in terms of our breathing, and as we control oxygen and carbon dioxide levels, those carbon dioxide levels affect pH. So in fact, if we change our breathing, it can affect our pH. Now, for the most part, respiratory system is focused on maintaining oxygen levels and carbon dioxide levels. But because part of that affects pH, you can have kind of this secondary effect where, yeah, as long as we're getting enough oxygen still, you can tweak your breathing rate to affect your carbon dioxide levels to also help out with pH. And in fact, you may see that as a symptom as well in terms of your patients. If they are going into acidosis or alkalosis, you may see their breathing rate change. You're like, why? You're not exercising or whatever. Why are you breathing so fast? It could be that the respiratory system is trying to help control body pH. Okay? And then lastly, we have renal mechanisms. What does renal mean? Kidney. So obviously, as we just got done with urinary system, we know that the kidneys are very effective at reabsorption and secretion, including hydrogen ions and also reabsorbing these bicarbonate ions. So it is also effective in terms of helping to control pH. So we're going to talk about just those 3 systems in a little bit more detail. But before we do, we have to remind ourselves of what we said, what makes an acid and what makes a base, right? Whether we are freeing hydrogen ions or binding them up. But what's the difference between a strong and a weak acid? So if we've got something like a very strong acid as seen on the left, that's like hydrochloric acid, what happens is if you put it into a solution, if you put it into water, basically all of those molecules, those acid molecules, fall apart and release hydrogen ions. So that has a big effect on pH, so it's a strong acid. Meanwhile, if we look over on the right-hand side there, that is carbonic acid. And I was going to point over there, but that screen still isn't working. We have carbonic acid, and carbonic acid, notice if we put it in that polar solution in water, some of them fall apart and release hydrogen ions, but some of them still stay together. So that's a weaker acid, has less of an effect on pH. Okay? So that's the difference between a strong or weak acid, is how well those ions as part of that molecule dissociate or fall apart. All right. So in terms of these 3 systems and how we can control pH, whether it's strong or weak acids, we have firstly our chemical buffer systems. So what we're doing here is we're just listing examples of different chemicals that we have in the body that can at times bind up or release hydrogen ions. So the 3 main chemical buffer systems that we have in the body, the first one is that bicarbonate buffer system. So the same one that we saw occurring in the respiratory system, in red blood cells, occurring in the wall of the stomach in order to make hydrochloric acid, occurring in the wall of like the proximal convoluted tubule in the kidney in order to allow us to secrete hydrogen ions, that system exists in many cells of the body. And by using carbonic anhydrase, we can affect pH by binding up that hydrogen and coupling it with the bicarbonate or doing the reverse of that. And so that bicarbonate buffer system is found in many different cells of the body. So it counts as part of our chemical buffer systems, even though we would also say it's part of the respiratory mechanism and the renal mechanism. Okay? In addition, we have a phosphate buffer system. So what you see here, there certainly is another molecule. We said calcium phosphate, remember, when we're talking about bone tissue and whatever. But we can see this one is phosphoric acid. So instead of 1 calcium with a +2 charge, we can attach 2 hydrogen in order to balance out that phosphate, if you will. And that is, again, a case where we can release a hydrogen ion or we can bind it up. So that's something else that can be phosphate is often found like inside of cells, and so that helps to control the intracellular pH. Then we have the protein buffer system. The protein buffer system, that's a whole bunch of different proteins. There are certain proteins in the body that can help with pH and help to bind up hydrogen ions as well. A main example of that protein buffer system is going to be like hemoglobin, right? Hemoglobin is a big molecule that can help to bind up hydrogen ions as necessary. Okay, so questions on that? It's called a chemical buffer system. It utilizes different chemicals around the body and different cells to very, very quickly control pH, bind up hydrogen or release it. So this is almost instantaneous. As long as we have these molecules around in that same fluid, they can do that as soon as they show up. Okay? So it's the fastest of our three mechanisms, and it involves different chemicals, whether they be, chemical buffers or proteins or something like that. Alrighty. Second one had to do with a respiratory mechanism. This has to do with, again, that same equation that we talked about before, the carbonic acid, where we're converting bicarbonate and hydrogen ions into water and carbon dioxide. And this equation, remember, can go either way. So if we need to bind up hydrogen ions, we might run it back to the left, right? And that would make it less acidic. If we need to release hydrogen ions, we would run it to the right. We would combine carbon dioxide with water, briefly making carbonic acid. That would release a hydrogen ion, right? Now, oftentimes that bicarbonate ion gets whisked away and put in another area, like in fluid, like the blood or something like that, and the hydrogen ion stays behind and makes that particular cell or something like that more acidic temporarily. But that is affected by how much carbon dioxide we have, right? So if you are breathing slowly, let's say, then carbon dioxide levels are going to increase. And with this equation, it's kind of like a seesaw, right? If we pile up more things on this side of the equation, then it tips it over that way, and we start releasing hydrogen ions. So the more carbon dioxide we have in the system, the more it's going to be converted into bicarbonate ions, and the more hydrogen ions we're going to be released, the more acidic it's going to become. The lower the carbon dioxide in the system, this equation is going to dip back the other way. It's going to tip this way, and we're going to, as we've gotten rid of carbon dioxide, we're going to need to make more. So we're going to combine hydrogen and bicarbonate in order to create more carbon dioxide and water, right? So if you're breathing rapidly or something like that, and you're getting rid of a lot of carbon dioxide, we might go the other way because your tissues might be becoming too alkaline instead. So again, with the respiratory system itself, it's primarily focused on oxygen and carbon dioxide. So it can't just change your breathing and focus only on pH, because its role is to make sure our oxygen levels and carbon dioxide levels are within homeostatic limits, but it can help with it. It can affect it. So like we said, in essence, those carbon dioxide levels are going to affect this. So when we are doing different breathing rates, right, if you remember, when we're looking at carbon dioxide and oxygen, we had like hypo and hypercapnia. Hypercapnia is where we have higher than normal levels carbon dioxide, or if we have that higher than normal hydrogen ions and it's becoming too acidic, then we may do deeper and more rapid breathing to get rid of some of that carbon dioxide and tip that equation back the other way, okay? On the other hand, so that's when we're going into acidosis, right? Acidosis is too much hydrogen ions. Alkalosis is the opposite, so we might see the lungs respond accordingly, where we have slower and shallower breathing. Shallower means we're not going to be exchanging as much at the alveoli, and that means there's going to be an accumulation of carbon dioxide in the blood, and that's going to then try to make it a little bit more acidic and help to neutralize things. Now, on the flip side of it, right, while the respiratory system, while the lungs, if you will, can help with pH, if the lungs aren't working properly, it can throw off pH. And we call that respiratory acidosis or respiratory alkalosis. We've entered into becoming too acidic or too basic, and the lungs are the problem. So rather than helping, they're causing the issue. Okay? And we'll talk about that a little bit towards the end. All right, and then finally, that third mechanism is the renal mechanism, right? So this is where, again, like we just got done learning about, the nephron is able to help control pH. Now notice that with our chemical buffer system as well as our respiratory system, what we're really doing there is temporarily binding up hydrogen or releasing it, okay? So we didn't actually change the hydrogen concentration overall. We just incorporated it into a big molecule, or we let it out on its own to float around as an ion. It's there, but it's just in a different form. In the kidneys, that's the only place and the only way that we can actually totally push out and get rid of a hydrogen ion. Because when we secrete things into the urine, it's going out, right? It's going down to the bladder, going out the urethra, and now it's exited the body. So that is a very effective way of controlling pH. We get rid of those excess hydrogen ions as opposed to just binding them up temporarily, okay? Now, the thing about it is, we know that the urinary system, while it's good at its job, it takes some time. Filtration, reabsorption, secretion, all of that stuff takes a bit of time, right, for it to run through the system. So overall, it's kind of the slowest of our mechanisms, but at the same time, it can be the most effective because, like we said, it's the only way that we can truly get rid of those different acids out of the body. So when we're looking at it, and let me get to a figure here, the key aspect of it is that we are secreting hydrogen ions, but a bonus of it is that we are often reabsorbing a bicarbonate ion, right? So if we're using that mechanism like the carbonic acid, and we're splitting it, and we are secreting the hydrogen ion into the urine, so now it's gone, what about that bicarbonate ion? We're still retaining that, and now that can go and bind up another hydrogen ion. So it's a twofer. Not only did we get rid of a hydrogen ion, but now we have an additional thing that is available to bind up a second hydrogen ion. Okay? So that's what we'll see when we take a look at it. And mostly that occurs, we know, in the proximal convoluted tubule, right? Because that's the first area where we're doing a lot of those tweaks. And what we're seeing here is a zoom up of that filtrate of that urine in like the proximal convoluted tubule. So here's our urine that's going out, and what you can see is that the walls inside of the proximal convoluted tubules, right, along making up its wall, have that same enzyme. So they have carbonic anhydrase inside of it, just like the red blood cells do. And what does that do? It combines carbon dioxide and water, and it can split it into a hydrogen ion and a bicarbonate ion. And you can see how the hydrogen ion will be then secreted, and the bicarbonate ion will be reabsorbed and put back into the blood. So that's our twofer, right? Now there is some kind of, when we put hydrogen ions here into the urine, that can make it more acidic. And that's why when we talk about the pH, there's a pretty wide range of pHs that are considered normal. Like when we were talking about the chemical sticks, the multi-sticks and stuff like that, it's like pH 4 to pH 8 almost would be considered normal based on what you're eating and what you're drinking and what your metabolism is. Your urinary system, your kidneys are going to be trying to balance that out, get rid of those acids or whatever, right? So if you're drinking, instead of regular water, you're drinking lemonade all the time. Anything tangy is acidic, right? Citric acid, a lot of citric acid in there. You're ingesting a bunch of citric acid, you're making your tissues more acidic, guaranteed your urine is going to be more acidic during that process. That increase in ingestion of acids is going to be handled by the urinary system. So in order to make it not too acidic, we don't want to be burning our urethras, right? If it hurts while you pee, there's something wrong, right? So you can see that we do bind up that hydrogen ion again. There sometimes are bicarbonate ions even in the urine, and we can then kind of neutralize it by binding up the hydrogen in the urine and making that water again. Then the carbon dioxide gets reabsorbed by those tubular cells again. But for the most part, what we're seeing here is the way that the kidneys are able to secrete hydrogen ions and reabsorb bicarbonate ions. And that is very effective in terms of controlling pH. This is another mechanism that we see here. Same thing, we're looking at, in this case, the collecting duct. Remember that the collecting duct is our last part of the kidney, where we are able to tweak that filtrate, just like we talked about the large intestine being the last part of the digestive system to do that. So in this case, we can do the same thing. We can do a little bit more secretion of hydrogen ions here and a little bit more reabsorption of bicarbonate here as well. It's the same process as occurring in what are called type A cells in the walls of the collecting tubule. And the same thing we do, we kind of bind it up in the urine so it's not burning our urethra, etc., or burning any of our other tubules. But we're getting rid of a hydrogen ion, and we are reabsorbing a bicarbonate ion. So it's a twofer in terms of controlling pH. Okay? And this is finally another way that we're doing the same thing, but here we see that we're helping to get rid of ammonia. So we do a secretion of ammonia, and when we get rid of that ammonium ion from the products from protein digestion, when we get rid of the ammonium ion, that process there actually allows us to conserve a bicarbonate ion as well. So even in this process of helping to get rid of metabolic waste products, it can have an effect on pH. Okay? And this is all happening within the kidneys. This is back in the proximal convoluted tubule again. All right. So at the very end of this, when we're talking about pH, we do mention a couple of situations where, again, we may get into a case where we're out of homeostatic range. So alkalosis and acidosis, we need to know those terms, okay? And in terms of how we can get into acidosis and alkalosis, there's a few different ways. We know we have three mechanisms, right? Chemical buffers, respiratory mechanism, renal mechanism. But if any of those are having an issue, the other two of the three are going to have to compensate. So if the respiratory system, we're not breathing properly, we've got pneumothorax, or we've got pneumonia or something like that, we're not able to exchange gases very well because there's a buildup of mucus and an infection, etc., then we may go into a problem of like respiratory acidosis, right? Something like that. So problems with our respiratory system can lead us into respiratory alkalosis, respiratory acidosis. So what we find is for respiratory acidosis, what's happening is if a person is breathing very shallowly, right, they're not doing exchange down at the alveoli very well, or if the gas exchange is hampered, like we said, in various ways, then carbon dioxide is going to accumulate. If instead the respiratory system is doing too much, like in hyperventilation, if you're hyperventilating, then that can lead to respiratory alkalosis. Because what's happening there is you are getting rid of excess carbon dioxide. So the amount of carbon dioxide that's being produced by your body is not increasing, but you're breathing as if it is. And you're pulling more carbon dioxide out of the blood than what the body needs. And so that's leading to alkalosis instead. Okay? So you're not matching the bodily needs with the respiratory function. So that's why, in fact, when you think about it, right, if you're hyperventilating, so let's say Saturday you're about to take your exam and you're starting to panic. Not you guys, because you are going to be well prepared, but somebody, right? What is one of the main things that they tell you to do if you're hyperventilating? Breathe into a bag, right? So you're breathing into a bag. What's that going to do? So number one, it gives you an audio cue. It gives you a feedback mechanism, more awareness about what's going on, right? There's all kinds of intentional behaviors that you do now to help be more aware about what your body's doing and help control it, right? So there's a voluntary aspect of it. So you hear that bag going, you're like, oh my gosh, that's too fast. So let's focus on, slowing it down. I can hear it, that bag opening and closing. But in addition to that, it encourages you to take bigger breaths, right, to really fill that bag. So you're not doing that shallow breathing. So now you're going to be doing a better external respiration or exchange. And when you're breathing into the bag, as you exhale, we know that that volume of air has a higher concentration of carbon dioxide than fresh air. So then as you breathe that back in, you're breathing in more carbon dioxide. In hyperventilation, you were getting rid of too much. So now we're actually recirculating it and we're bringing it back in. So that's also going to help to restore our carbon dioxide levels and our pH. right? Okay. Another way that we enter into acidosis or alkalosis is called metabolic acidosis or metabolic alkalosis. This can have to do with a problem in the kidney. So if there is a kidney malfunction, that's certainly going to contribute to it. But in addition, the ingestion, right, as part of the digestive system, if you are ingesting things that are extremely acidic or extremely basic, then the body's going to have to deal with that. So metabolic acidosis, too much of things like alcohol. So in the kind of detoxification process for alcohol, that can make things acidic. Or if you're losing bicarbonate ions as well. So for instance, taking in acids, if the circulation is poor, so you're not kind of perfusing your tissues and things like that, things can build up. Ketosis, if we are in a starvation state and we're secreting a lot of ketones, or creating a lot of ketones because we're breaking down fats, etc., then you get more fatty acids. That's making it more acidic. And then, like we said, kidney failure. So these types of things can make your overall systems more acidic. In terms of metabolic alkalosis, again, it can come from kidney malfunction, but it also can come from either taking in a bunch of alkaline things. So there are a lot of dark leafy green vegetables that are fairly alkaline. So if you're a vegetarian and you're taking in a lot of those in your diet, more so than other people, your body's going to have to adjust for that. In addition, vomiting. So vomiting, if you're vomiting, you are getting rid of stomach acid. That is all part of the pH balance in the body. You're supposed to be secreting stomach acid, mixing it with your food, and then helping to reabsorb some, neutralize that, like we saw before. Neutralize it with the duodenal gland secretions and the pancreatic secretions, stuff like that. If there isn't as much hydrochloric acid coming down, when we release from the pancreas and the duodenum, now it's actually making it too alkaline. It's making it too basic because we don't have that acid to balance it out, right? So vomiting can put you into metabolic alkalosis, and then constipation as well. Because as we're migrating through that large intestine, right, it's just going to do its job. It's going to be reabsorbing water, and it's going to be reabsorbing some ions, including bicarbonate ions. And the longer you leave it in there, the more it's going to do its job. And so that can also lead to metabolic alkalosis. Okay. So in essence, like we were talking about, there are ways that we can get out of balance with three different mechanisms. If one of them is the problem, if the kidneys are the problem, then the respiratory system and the chemical buffers are going to try to compensate. If the respiratory system is the problem, then our chemical buffers and our kidneys are going to work harder to try to compensate. Okay? So that's kind of how we have those ways of the body having redundancy. Like we have two kidneys, we can kind of survive without a kidney. We have three different ways that we can help to control that pH in the body. Okay, so changing our breathing rate can certainly help with that. Changing, like I said, our filtration, our secretion, and things like that in the kidneys can happen as well. Okay? All right. So this is just basically saying that those activities that we just described those organs doing, as the kidneys are working, as the lungs are working and stuff like that, can help to bring it back into a normal level. All right. So that finishes up that chapter on fluid balance, electrolyte balance, and now pH. Okay? Questions? So key aspects of that is we have to define pH. We have to know what it is. We have to talk about, okay, so what's an acid, what's a base, what makes it a strong acid, what makes it a weak acid, etc. And then we need to know that there are three ways of controlling it. We need to know the chemical buffer system, our renal mechanisms, and our respiratory mechanisms. Be able to describe how each of those can help to control pH. What does that do to our hydrogen ion concentrations, etc. Okay? So as I mentioned, luckily this chapter is nothing new. I mean, mostly it's review of either things you learned in chemistry or things that we learned in other systems of the body. How these systems are working together to maintain a stable internal environment, right? Whether it's fluids, whether it's solutes, whether it's our acids and bases as well. Okay. So now, with the end of the new material, the question is, what would you like to review in terms of the exam? We have digestive system, we have urinary system, and we have this fluid and acid-base chapter. They may have pressing questions that they wanted to answer, topics that they wanted to go through. Let me bring up digestive system slides as well. Yeah, Angie.
I don't know, it's just, you have in bold number 22 in digestive system, explain the role of the appendix. Like, the only thing I saw was like it has the flora on balance, right? But like, I don't know how.
Yes.
Folded, but that's all I know.
That's a good question. I wonder if like I got a little crazy in the bolding and just ended up hooking that one in there. I'm thinking maybe it's an editorial thing. But you're right? For the most part, our appendix is immune system, right? So we've got within its walls lymphatic tissue that are protecting us against pathogenic bacteria, etc. But the other aspect of it is that it does house beneficial symbiotic bacteria to maintain that population within the colon. And those beneficial bacteria do help, right? They help to give us vitamin K, help to break down other carbohydrates that we can't, etc., and ward off our pathogenic ones. So the only way that I would see that coming into, off the top of my head, coming into a short answer question is if we were kind of going through and we were saying, okay, so what are the different organs of the digestive system, and kind of describe what each one does in terms of benefiting the digestive system. And so in a way, appendix is part of the digestive system, or at least attached to that cecum there. So maybe as an option, if you were listing organs, you could say, well, that's in there, but I don't foresee a big short answer question focusing on the appendix. Yeah. I just threw it in there just to see if you guys are reading it closely. Yeah. Right? Other things. Yeah.
On the urinary system,
Mm-hmm.
I have a couple of them.
Okay.
I think it's number 8. It says, explain what tubular secretion is.
Yeah.
And then it breaks down. It says, what is secreted, how are they secreted, and that's where I'm stuck. Is it transport proteins?
Right. So that's a good point. So that type of question where we're asking at a cellular and molecular level, how is the system doing its job, right? Because that's what it comes down to. That's what we're finding out. Respiratory system, urinary system, digestive system, cardiovascular system. We get down to the cells that make up these organs, and inside of those cells, that's where all the magic is happening. That's where we're creating hydrochloric acid. That's where we're reabsorbing or secreting ions from the filtrate in the kidneys, etc. So, point being is that when we're talking about urinary system, and we're talking about its 3 processes, we needed to be able to describe the details of each one. Now, filtration was a big one, so I'm throwing some extra information in there, just reminding us of other things that we want to review. Filtration, secretion, and reabsorption. Filtration of our 3 processes was our biggest one, right? Just because there's a lot of control aspects of filtration as well, where we're like, oh, dilating, constricting the afferent and the efferent arteriole, juxtaglomerular apparatus. We have this whole checkpoint. What's that about? What does that do? That's all about filtration, right? Are we filtering too fast? Is the blood pressure too high? Things like that. And do we need to make adjustments? By the way, juxtaglomerular apparatus is like, I think, a separate question, right? It'd be, okay, tell me about the juxtaglomerular apparatus. What are the cells involved? What are they doing? What's the whole point of it? That could be a 5-point question right there, because it's kind of complex, right? But anyway, is it not in there? I don't know. We'll come back to it.
I'm not seeing it anywhere, so.
Okay.
That doesn't mean it's not there.
So, point is, when we're talking about these processes, we want to be able to say specifically, we want to say what it is. Whether it's a digestive process like ingestion or absorption or secretion, what is it? Be able to define it. So for secretion, right off the bat, we'd say, okay, secretion is where we are moving a solute from the blood into the tubule, right? So we have to define it and be able to tell the difference between that and reabsorption. Second thing is we got to be able to say in the nephron, just like the digestive system, we have all these different parts that do different jobs. The nephron, we have different parts too, and each one has a different job to do. So we got to say, okay, for secretion, where does secretion happen? And luckily, as we were finishing up that fluid and acid-base chapter, we talked about some of the primary things that are secreted: hydrogen ions and then potassium ions, because the sodium-potassium pumps and things like that. So the primary places where secretion happens are going to be the proximal convoluted tubule. That's your go-to for the nephron, right? Most everything happens there in terms of secretion and reabsorption. That's why we have the microvilli and all that type of thing. And then it also occurs in the collecting tubule, like we saw. We saw secretion of hydrogen ions in the collecting tubule. So that's another place that you could say.
Also in the distal, right?
In the distal, you do get some, right? Yeah. So it's kind of, we're starting to get towards that cleanup crew. Like, hey, do we need to do a little bit more reabsorption, a little bit more secretion, etc. And then the mechanism. So in terms of the mechanism, we have to be able to say, how is that ion or molecule or whatever, how is it moved into the cell or into the bloodstream or out of the bloodstream or whatever? For the most part, with our secretion, it has to do with active transport. Active transport is going to be pumping it, right, pushing it from one side of the membrane to the other, because it's going against the gradient. And it's like, well, it's so important, we got to get it out of here, so we're going to be secreting it. Sodium, potassium pump, sodium, hydrogen pump, etc. Those are the ways that we're kind of pushing across there. So active transport, that's the roundabout answer, is your main mechanism for doing secretion, right? So you just say active transport. For our other things, when we're doing reabsorption, some of those are just diffusion. Some of them will come back on their own according to concentration gradient. Some of them are going to be like a facilitated diffusion, where we have to have a carrier molecule. Like for glucose, we have to have a carrier molecule to get it across. There's a concentration gradient, but we've got to escort it across. So facilitated diffusion, or what we call it like a symporter, where we're bringing in sodium and glucose, or sodium and amino acids, right? So we have facilitated diffusion, and then of course when we're reabsorbing water, what's the main way, the mechanism for water reabsorption? Aquaporins, and it's osmosis. Osmosis drives the process, and it moves through those aquaporins. It can move through cells as well. It is, our cell membranes are permeable, somewhat permeable to water, but they're more permeable when we have those little aquaporins inserted in there, okay? So you gotta have those mechanisms in there too. And as a reminder, let's then apply that to what we saw in digestive system. In digestive system, we have those 7 processes. We were already talking about that. We need to know what they are and where they occur, okay? But a key aspect and a more detailed aspect of the digestive system that we talked about were all of those nutrient macromolecules and how we break them down. That was a big part of digestive system. So carbohydrates, proteins, lipids, nucleic acids. We need to know those 4 nutrient molecules. Those are the big macromolecules. We got to talk about, okay, what's the form that they occur in that's more complex? What's the simplified form that we get after we've digested them, chemically digested them, and what's the enzyme that's going to do that? Part of that, the last step in that process, and we reviewed that at the very end of the digestive system chapter, is, again, how do we get those to come into the body? How do we absorb those? What's the specific mechanism? And in many cases, we're still talking about things like facilitated diffusion for things like glucose, and maybe even some active transport, osmosis for water, stuff like that, right? So for carbohydrates, for proteins, for our lipids, and for our nucleic acids, for each one of those 4, you want to review that. And I'm pretty sure, and again, you'll have to double check me, but I'm pretty sure, right, you could get a short answer question where it just focuses on, like, proteins. Okay, tell me, what's the complex form? That's an easy one. Protein is the biggest complex form, right? But the products are going to be our amino acids and even maybe our peptides, or short peptide chains. Those are the products. That's the simpler form. So we need to know the complex form and the simpler form. What's an enzyme that breaks down a protein? Pepsin's one of them from the stomach. We have proteolytic enzymes coming from the pancreas, right? So you have to be able to give an example of, okay, what's an enzyme that chemically breaks down this organic molecule? Where does it come from? Pepsin's coming from the stomach. pancreatic proteolytic enzymes coming from the pancreas, right? You got to be specific enough. What's the enzyme? Where's it coming from? And then, like we said, last step in that is like, how do we absorb it across the body wall? Is it like a facilitate diffusion for a lot of those? Yep. That's the way that we actually absorb it. And that's happening in that small intestine. So there's a story there for the digestion. Digestive system is about breaking down big things to small things. And the things that we're talking about are, in many cases, macromolecules, organic nutrient molecules that we need to make bigger to smaller. And that process is chemical digestion, as opposed to physical digestion, where we're mashing things up. That's digestion too, but we're not breaking chemical bonds. Chemical digestion is going to involve chemicals, in most cases enzymes, that are breaking those chemical bonds and giving us those simplified products, right? Or monomers and stuff like that. So for each of those, you could write up like a flashcard or just like a little, summary or a little table for yourself. What's the macromolecule? What's the enzyme? Where does it come from? What's the product? How do we absorb it once we break it down, right? Carbohydrates, you're talking about polysaccharides, going down to things like disaccharides and monosaccharides, right? And so you have things like amylose as one of your major complex carbohydrates, and amylase, salivary amylase, pancreatic amylase, are going to be the ones that are helping to break that down. And then, of course, we have our lipids, right? So we have various lipids. Triglycerides were our main example for our macromolecule, breaking it down to monoglycerides and glycerol. And then we had, remember, things like lipase, right? Lipase, lingual lipase, gastric lipase, a little bit, pancreatic lipase, big one. Just a small addition to that, remember that we talked about brush border enzymes, right? So the cells lining the small intestine have microvilli too. It increases surface area, one of the ways we increase surface area, and it looks like a brush, and there are enzymes embedded right into those microvilli, and they're the last step of chopping it small enough to bring it across the body wall. So brush border enzymes do handle things like proteins and lipids and carbohydrates as well, kind of like the pancreas. And there's nucleic acids and nucleases and stuff like that. Okay?
Brush border enzymes, it's just a generic term. We didn't go into like.
Right, we didn't go into specific ones.
We just have to know that that's an example of one of the ways.
Yeah. Okay? So for those short answer questions, you want to make sure that you have a detailed example to give if asked. Again, I don't ask you every single thing usually. If we're talking about processes of the digestive system, I'm not asking you all 7, I'm saying give me 5, describe them, tell where they occur and stuff like that. If we're talking about the chemical digestion, I'm not asking you, tell me proteins and lipids and carbohydrates. I'm usually asking you 1, right? There's enough information for one of those to give you 5 points for a short answer question. So just make sure you have enough detail. Like, what's the name of the enzyme? Where does it occur? What's the complex form? What's the simplified form? The subunit, stuff like that. Okay? Yeah.
Another question on the urinary. One of the questions, it's not bolded, but it just says, identify the components of the nephron. One of my questions was, is the glomerulus considered part of the nephron?
Yeah, so the glomerulus itself is considered part of the nephron because when we talk about the 3 processes, the filtration, the reabsorption, secretion, it's a critical component. We have the glomerulus and then we have the glomerular capsule around it, and both are going to be critical. That's where filtration is occurring. The capsule is going to be catching that filtrate that's going to become urine. So that's kind of the beginning of our nephron. Then we have our proximal convoluted tubule and our descending limb of Lupus-Pendley, the ascending limb, distal convoluted tubule, and then technically that's where our nephron ends. But they do jump into a collecting tubule where, like the large intestine, we have this last chance to be secreting and reabsorbing, stuff like that. And that's where a lot of our hormonal control, where it's like, do we need some more? Here's a signal. Yep. Pick up that out of the filtrate before we lose it. Whether it's water, whether it's sodium, things like that. Okay? Thinking about urinary system as well, when we're talking about water reabsorption, the whole idea of the kidneys, not the whole idea, one of the main ideas of the kidneys is that we are controlling fluid levels in the body. Not only solutes, the concentration of our fluids, but the solvent, the water as well. And we said that the water reabsorption that occurs in two different ways, right? We have facultative water reabsorption and we have obligatory water reabsorption. So you need to be able to describe, okay, well, there's two types. What does that mean in terms of obligatory? We have to, we're obliged to. Facultative, optional. That's later on, like we said, in the distal convoluted tubule and the collecting tubule, where ADH comes into play. Where if we need to reabsorb water, we have this optional extra amount of water we can reabsorb, like an extra 10 to 15% that we can pull back in, as opposed to the obligatory water reabsorption. That's right in the proximal convoluted tubule and the ascending loop of Henle. We're always pulling that 80 to 85% back in. We have to get it back so we're not a dried up husk, right? So talking about how the kidney and the nephron control water secretion, not really secretion, but water, letting water go out of the body or reabsorbing it back, and water balances in the body, that is itself kind of important, right? Yeah. The whole nephron loop and the collecting tubule, that's a topic, right? That whole vertical osmotic gradient there. The nephron itself, the nephron loop, creates the gradient, right? The nephron loop creates that 300 to 1200 milliosmoles from the cortex down to the medulla. It does so by reabsorbing sodium, active transport of sodium off of the descending limb, which draws water off of the ascending limb, okay? So that loop creates the gradient, and that allows that collecting tubule to use the gradient, to reabsorb water as necessary. It creates an osmotic gradient so that if we need to reabsorb water, we release antidiuretic hormone, we insert all those aquaporins, and the water will use the aquaporins. Because if we don't have a concentration gradient, then the water's not going to go. It has to have a gradient between inside the tubule and outside the tubule, so that when you open up those channels, it goes, oh, there's a whole bunch of me water here, there's not as much of me over there, so I'm going to move out of the tubule and go back ultimately into the bloodstream. Yeah, Angie.
Number 5, what remains behind in the blood? Was that like the blood cells, some glucose, and then was that it, or I thought there was a couple other things?
So that's a good point as well. When we're talking about the 3 processes in the urinary system, we need to be able to describe each of the 3 processes. We need to say specifically where they occur. You can't expect full credit if you just say, hey, you know, the kidney, it filters stuff. Yes, it's true, but that's like a 4th grader definition of that, right? So what you guys as aspiring health science professionals know is that filtration occurs only in the glomerulus, right? So in that one specific part of the nephrons where filtration occurs. Then when we're talking about it, in each of those 3 processes, we say, okay, what are substances that are going to be moved according to this process? So with filtration, we said that it's size selective, right? It's like a colander, which did you guys see in the eclipse, all those people using colanders in order to visualize the shadow of the eclipse? Actually, I hadn't seen that before, or if I saw it before, I forgot it. But each of those little holes act like a pinhole camera, and so you hold the colander and the light comes down and you see like the little crescent moon shape of the eclipse down on the ground. It's kind of cute. Point is, a colander, like that glomerulus, only lets small things go through, right? So if you have a colander, the pasta is going to stay in the colander, the salt and the water are going to drop through the colander. So when we're talking about filtration, things that are going to stay in the blood are going to be the big things: red blood cells, white blood cells, platelets, large molecules like proteins, like albumin, like antibodies, things like that. Those are going to be some of your examples that you would say for things that would stay in the blood. Things that would actually be filtered: water, ions. You can name different ions: sodium ions, potassium ions, calcium ions, whatever. Some hormones are small enough to slip out. Some digestive products, glucose, amino acids, stuff like that, those slip out as well. Those are all examples of small things that would be filtered. And then the last part of that story, if you will, about filtration is saying, okay, what's the force behind it? What drives these things to be filtered? And in this case, it's just straight up blood pressure, also known as hydrostatic pressure. that fluid pressure of pushing these things through the filtration membrane. It's just that there's more blood arriving there all the time. The afferent arteriole is bigger than the efferent arteriole. Can't get out fast enough, so let's squeeze through those holes in the wall.
Do we have to know those numbers that go back within that glomerulus?
Good point. So Andy was asking about, okay, so when we were talking about pressure, we said like 55 millimeters of mercury in here, blood pressure, versus you don't have to, like when we were doing respiratory system, you don't have to know actual numbers. Just know that we have a higher blood pressure there in those glomerular capillaries, and it is higher than all of the other forces, right, in millimeters of mercury trying to push stuff back in. It's overwhelming those, so that our net movement is always out. We're always filtering. But you don't have to know those numbers. Yeah. So in that case, then, right, the driving force that causes filtration is blood pressure. Okay? Okay. One of the other things that I would highlight in those two chapters, and granted, right, we're looking at it, digestive system and urinary system chapters were bigger than this chapter having to do with fluid and acid base. So you would expect probably more questions from those chapters just because there's more information there, right? So it's not going to be exactly divided into thirds per se. But when we did digestive system and when we did urinary system, one of the other topics we went through was, okay, how is this system controlled and coordinated, right? And it's going to be that same answer. There's going to be two types of extrinsic control, neural control and hormonal control. And then there's some sort of local intrinsic control as well. So that applies both to urinary system and to digestive system. Talking about how this system is controlled and coordinated, and talking about the extrinsic and intrinsic control mechanisms. Right? So for digestive system, neural control is going to be autonomic nervous system. For urinary system, guess what? It's going to be autonomic nervous system, right? Because autonomic is automatic. It's doing it at a subconscious level and making sure these things are performing at an appropriate level for the body's needs. So sympathetic and parasympathetic nerve fibers going down and telling blood vessels to constrict or dilate, or telling glands to release more secretions, or increase the muscular contractions of that smooth muscle in the wall of the alimentary canal and stuff like that, right? Now remember, for digestive system, parasympathetic is your big one. Parasympathetic is rest and digest. So it's parasympathetic neurons that are going down and telling these different organs of the digestive system to increase their activity. Yeah? So that's neuroextrinsic. Extrinsic hormonal, you have to have at least a couple examples of hormones that affect that system. So for our digestive system, we said there's gastrin coming from the stomach. So that's kind of ironic because it comes from the stomach, and then technically it's a hormone and it mainly affects the stomach. Still counts, though, as a hormone. We had CCK, cholecystokinin, and we had secretin. And then we also talked about GPL1, kind of one of the newer ones when we were talking about Ozempic and stuff like that. So those are hormones that also then tell the organs, they go into the bloodstream, but they tell the organs to increase or decrease their activity. So be able to, kind of name at least one of those as a hormone and say what it does. For urinary system, hormonal control exists as well, right? Antidiuretic hormone, aldosterone, those are hormones that affect urinary function. And then we have the intrinsic control system. So our third local control for the digestive system is the enteric nervous system. So a neural network that goes between different sections of the alimentary canal, oftentimes from the duodenum to different other sections. Because the duodenum is the beginning of where we're really going to be doing digestive, like chemical digestion and absorption and stuff like that, right? So it needs to communicate with other sections along the alimentary canal, like back to the stomach. It needs to communicate to other organs of the system to make sure that they are acting in concert with one another. So the enteric nervous system are neurons that connect these different organs and produce the local reflexes that communicate between them. So it's a within system kind of communication system. So that's enteric nervous system. And then for urinary system, our local system technically is the juxtaglomerular apparatus, right? Now, juxtaglomerular apparatus, again, is kind of straddles the line of extrinsic, intrinsic. It comes from the kidney. That RAS mechanism with the renin, angiotensin, ultimately aldosterone system, right? It starts with renin being released from those juxtaglomerular cells. So it's a little bit involved in our extrinsic control. But at the same time, we also have that what's called the myogenic response, right? The myogenic response, myo means muscle, that afferent arterial, if for some reason we get a big blood pressure increase from the body or our blood volume goes up all of a sudden, those afferent arterioles are going to stretch out and they're going to do this reflexive constriction to make sure that we're not blowing out those glomerular capillaries that are just downstream of it. So that is a local response. It's like, hey, I don't know what you guys got going on over there. There's something big happening. Don't ruin my day by blowing out my kidneys, right? Don't blow out my glomeruli. So I'm going to protect them by automatically adjusting the afferent arterial diameter in response to, oh, am I being overstretched or whatever, okay? So those are, again, local controls, local changes that that organ makes that affects its function. So in both of those, that's usually kind of a topic as well, talking about how this system is controlled and coordinated to meet the needs of the body, extrinsically, intrinsically, etc. Okay? So when we're going through these, I know we're getting down, we got about 3 minutes or so, so I'll pipe down, kind of throwing stuff at you and see about addressing if you have a few other questions. But as we're going through these things, and as we're talking about this is a topic, that's a topic, that's a topic, make notes about those. I mean, you've got your exam objectives, which are effectively doing that, but make your notes about those and then create study materials for yourself on each of those topics, right? So talk about tubular secretion, tubular reabsorption, glomerular filtration, talk about the control systems of the digestive system, neurocontrol, hormonal control, enteric nervous system. Write that stuff down, get a nice compact answer for yourself, make it into a flashcard, and then practice with it, right? So that if you get questions related to that, short answer questions or multiple choice questions, like enteric nervous function does what? You're like, I don't know, is that even urinary? I don't remember, right? Creating those little study topics for yourself right now when they're fresh is going to be easier to kind of parse out your studying and focus on one thing at a time and make sure that you're covering those topics. Right? Okay. So, anything pressing in terms of other questions in our remaining minutes? Yeah?
The pancreas is exocrine.
Both.
And
Right. Yeah. Again, we're kind of straddling a line there. We're just getting into endocrine system in lab, and so we're learning that the pancreatic islets, those little bleach spots, are actually endocrine in function. And they release hormones like insulin and glucagon that ironically help to regulate glucose levels in the body. But the main part of the pancreas, those pancreatic acini that we see all over the place, those are the ones that are producing our pancreatic juice, right, with a lot of that sodium bicarbonate to neutralize the hydrochloric acid, and releasing our enzymes, our CCK, those are hormones. Our enzymes like pancreatic lipase, pancreatic amylase, and our proteolytic enzymes. So those are the ones that are primarily digestive in function. And those are exocrine because they dump into a tube that leads right to the duodenum and mixes it with the food. Yeah, so it's both. So it's interesting to find out, right? These organs that we're looking at that are primarily have this one role or one or two roles in a system, most of them also have an endocrine function. They have some sort of little cell somewhere in them that will release hormones that affect their activity or affect other organs and communicate to it as well. So technically, endocrine system almost involves everything in the body, but as we get to it after the next exam, we'll focus on the primary endocrine organs there. That's their main job. Other things? Okay. We're right at time there. So like usual, I've got office hours after lab today. And then tomorrow and Friday and stuff like that, I don't have scheduled office hours, but if you need me, you can send me emails. If you're developing these answers, you want to send them over and say, how's this for an answer for this question? I can always double check it, give you feedback on it and stuff like that before you really get studying it. Or if you want to meet online, right, Google Meet or Premium Cafe or whatever, I'm happy to meet as well. Okay? All righty.